Thanks for taking the time to hear my concerns about a bat research facility that's being built at Colorado State University on its Foothills campus in Fort Collins. Why are scientists so interested in studying bats anyway? Well, the reason is because their immune systems can harbor many infectious diseases asymptomatically, including dangerous ones like Ebola or Nipah. The idea of this research is that supposedly disease-free bats can be flown into the Denver airport and shipped to the Foothills campus, where they can be housed and bred so that cell lines can be developed and shipped to BSL-4 labs to be challenged with dangerous pathogens. But how do they know for sure that the bats are disease-free? Well, they try to determine the bats are disease-free using two methods, neither of which is foolproof. First, they'll use a PCR test to confirm the nucleotide sequences of the viruses that are associated with these infectious diseases and that they're not present in mucosal linings, feces, or urine of the bats. A negative PCR test does not mean the virus is not in the bat. It only means that there is not an active infection that's spreading the virus particles into the bat feces, urine, or mucus. Again, these bats can harbor the viruses asymptomatically in their body tissues, so the PCR test is not valid. Secondly, they'll use a blood test to determine if antibodies against the viruses are present. If antibodies are found, it means the bat has had an infection in the past or is currently infected, even if there are no symptoms. However, a negative blood test does not rule out the possibility of viruses being present and dormant in the bat. Here's the problem. When you keep these bats in closed environments for research, the possibility of viral mutations accelerating is high because they're not out in nature in their normal habitat that has buffers to prevent this abnormal acceleration. This drives new or novel virus emergence, and that's exactly what they're looking for. They call it virus spillover. They will tell you that they're not doing gain-of-function research, but they most certainly are. They are providing the opportunity for a new virus to emerge and see what functionality it might gain, including cross-species transmission. If something comes out of this from an undetected disease-harboring bat that can be transmitted in respiratory droplets between humans, we could have a disaster on our hands. Why? Because the Foothill Campus Lab is only a biosafety level 2, BSL-2 lab that would not be able to contain it. How do they keep getting away with doing this research? Well, because they have you convinced that by forcing a potential bioweapon to emerge, they can get ahead of it and develop a vaccine that would be prophylactic or protective. This is a loophole that is permitted by the Bioweapons Treaty of 1975. The treaty bans the development, production, and possession of biological weapons except for the production of prophylactic or protective vaccines. But there is no prophylaxis, i.e. prevention of infection, nor the ability to stop the spread in any injectable product called a vaccine. And that lesson should have been learned after the mass distribution of COVID vaccines. The reason is quite simple. The spread of these infectious diseases or bioweapons always comes through the epithelial barrier that separates the inside of the body from the outside. Now, these can be spread by inhaling aerosols or by swallowing something that comes in contact with the barriers. The protections against this happening are either on or in that barrier. The antibodies created by injectable vaccines are serum antibodies deep inside of you, not mucosal on the barrier, and cannot provide that protection, and that's a fact. Why do they do it then? The idea is that the serum antibodies will attach to proteins on the bioweapon after the bioweapon has gotten through the barrier. What proteins? The exact same proteins the bioweapon uses to attach to cellular receptors that are on the barrier. So what's in the vaccine? The weaponized part, or the message to make the weaponized part, so antibodies can be built against it. It must be the weaponized part, or the antibodies would not be effective at blocking cellular attachment. I hope you can see that. The problem is it takes a couple of weeks for antibodies to be built against what got injected. So those proteins are going to attach to cellular receptors on blood vessel walls, nerve tissue, and organ tissues inside the body. not on the epithelial barrier. And this causes all sorts of problems and pathologies that are listed on every single vaccine package insert that was ever made. So, what are we doing? We are allowing dangerous biological research on bats in closed quarters away from their normal habitat in BSL-2 labs close to human and animal populations that could easily escape, all so we can inject ourselves with a weaponized part of what escaped with the idea that that will protect us. None of this makes sense. What does make sense is government oversight to stop the dangerous research that drives this emergence of new pathogens.